Human body consists of huge number of cells. Each of them are having genetic material DNA. In human being, the haploid genome contains approximately 3 billion base pairs of DNA which are packed into 23 chromosomes. Of course, most of the cells, that is, somatic cells in the body are diploid with 46 chromosomes. That means 6 billion bases of DNA per cell is present in human beings. We know that the distance between each base pair is 3.4 angstrom or it is 0.34 nanometer. Therefore, if we calculate the length of the DNA in each cell, it will become 2 meter. Quite huge when compared to the size of the nucleus in which it must fit. Moreover, it is estimated that human body contains approximately 50 trillion cells. So, if we calculate the length of the DNA in all the cells, it will become 100 trillion meters of DNA per human. Quite interesting. Now, consider the fact that the Sun is 150 billion meters from Earth. That means each of us has enough DNA to go from Earth to the Sun and back more than 300 times. But such a huge molecule must fit into a very tiny cell organelle called nucleus, which measures around 5 to 6 micrometer. How it is possible for a huge DNA molecule to fit into a tiny nucleus? The answer to this question is that certain proteins compact the chromosomal DNA and fit it into the microscopic space of the nucleus. So, now let us learn how the DNA packaging occurs in the eukaryotic cell. Histone is a positively charged protein containing higher concentration of basic amino acids like lysine, arginine, and histidine. There are 5 classes of histone like H1, H2A, H2B, H3, and H4. The H1 histone is loosely attached to the DNA and it is called linker histone, while the others, that is H2A, H2B, H3 and H4 are called core histones. All histones except H1 are present in equimolar amounts and H1 is approximately half of the other. The packaging of DNA starts when the histone protein interact with DNA. 2 copies of H2A, H2B, H3 and H4 together forms a histone octamer. The double-stranded DNA wraps twice around the histone octamer. This super twisted helix forms a spherical particle of 10 nanometer diameter. It is called nucleosome. Each nucleosome consists of octamer and 146 base pairs of DNA. When H1 is also entering the reaction, 2 supercoil turns with 166 base pairs of DNA are produced. The function of the H1 is to stabilize the DNA. A short stretch of DNA connects 2 nucleosome assembly called linker DNA. The size of the linker DNA varies from one organism to another. Typically, it ranges from 20 to 60 base pairs. The observation by electron microscope revealed the fact that the nucleosome appear similar to beads on this fringe. DNA become more compact when nucleosome and linker DNA coils into DNA fibers. The packaging of DNA shortens the fiber length about 7 folds. In other words, the DNA, which is 1 meter long, will become just 14 centimeter long fiber. Despite of shortening, DNA is too long to fit into the nucleus. Therefore, nucleosomes are further supercoiled to form a structure called solenoid of 30 nanometer diameter. By this time, the DNA is folded to about 50 times, but still it is not sufficient to fit DNA into a nucleus. So, another group of proteins called scaffold proteins help in the further packaging of DNA. These scaffold proteins help in looping 30 nanometer fiber further and forms the chromatin fiber of 300 nanometer diameter. Finally, these chromatin fibers are further supercoiled and condensed to form chromatid. The chromatid attached with a centromere now become chromosome. Each chromosome has two sister chromatids. The chromatid contain two distinct regions in it. They are euchromatin and heterochromatin. Tightly packed DNA, which looks darker, are called heterochromatin. It is transcriptionally inactive, while loosely packed DNA, which looks lighter, are called euchromatin. It is transcriptionally active. So to conclude, DNA packaging is an important process in living cells. Without it, the cell is not able to accommodate large amount of DNA into its tiny nucleus.